On this episode of Nova Science Now, can a new DNA test tell us what diseases we'll get in our lifetime? They say it's easy as spit.
Spit.
That's right. Just fill up a container with saliva.
Took me 90 seconds.
Send it off to a lab and read your genetic future.
This is your actual genome.
And we want to do that across the entire population.
And one of these two paintings is a Van Gogh original worth millions. The other, a copy created by this artist for a Nova Science Now computer challenge.
Do you think she has a shot against the computers?
I think it will be a challenge, yeah.
3 teams of computer scientists
This white thing is showing through.
come up with programs to pick out our fake among 5 Van Gogh originals.
These are genuine Van Goghs.
Will this new technology work? The results may surprise you as much as they did us. Also, the scientists who built this machine say it can make like a tree.
We're trying to mimic what a tree can do, and these are the leaves of that tree.
And pull carbon dioxide right out of the air to reduce global warming. And where did the idea come from? His 12-year-old daughter's science project.
Hello.
We'll show you how, and more on this episode of Nova Science Now. Hi, I'm Neil deGrasse Tyson, your host of Nova Science Now. It seems like every week, scientists uncover more secrets of our DNA, revealing how our genetic code can help shape and influence our lives. And for some, this is scary, raising fears that insurance companies or employers might see our genetic profile and hold it against us. Well, Mr. deGrasse Tyson, I've seen your genetic profile, and it's not pretty. In fact, you're much too much of a genetic risk for us. But some say that knowing our genetic risk for disease will actually lead us to longer, healthier lives. Well, Neil, I've reviewed your genome. Here are my recommendations. And we prepared a bottle of vitamins, specially for you. Well, thank you. Live long and prosper. So, what can genetic testing actually tell us about our chances for a long and healthy life? And will that knowledge help us or hurt us? Can you imagine a simple test that tells you how healthy or not you'll be in the future? If you'll get cancer, gradually lose your memory, or be suddenly stricken by a heart attack? Sounds like something out of science fiction. But could this be possible today? Judging from the headlines, you might think so. New companies have just burst onto the scene, proclaiming the dawn of a new age of personalized genetic testing.
We're at the beginning of a medical revolution.
So this is where the DNA arrives.
Dietrich Stephan is a founder of Navigenics, which specializes in medical testing. For a fee of $2,500, they'll screen your DNA and tell you your genetic risk of getting a variety of scary diseases.
We are testing an individual to provide them a snapshot of how genetically loaded they are so that they can potentially reduce their risk, and we want to do that across the entire population.
The entire population getting their DNA scanned? It sounds a little like the movie Gattaca, where from birth, everyone's destiny is foretold by their genes.
Neurological condition, 60% probability. Manic depression, 42% probability. Attention deficit disorder, 89% probability. Heart disorder, 99% probability. Life expectancy, 30.2 years.
30 years.
I wondered what was lurking in my DNA, and Navigenics agreed to test me for free. There was just one thing I needed to do. Spit. Quick start guide.
You have agreed to provide us with a saliva sample. And
Spit.
It's spit.
Looks dangerous.
It's a couple of millimeters.
Biohazard.
Saliva is the most painless way to collect a sufficient sample.
So you're telling me you want me to spit into a cup?
Takes about 5 minutes.
5 minutes of spitting.
Correct.
Thinking of pastrami on rye with provolone, bag of potato chips. Luckily, it turned out I'm pretty good at salivating. Thinking caramel candy bar. Took me 90 seconds. My spit then traveled to a lab in California where they extracted and analyzed my DNA. DNA is made of long chains of 4 chemicals, best known by their initials, A, C, G, and T. There's about 6 billion of these letters in the human genome, but reading every one is painstaking and expensive. So the latest tests look for things called SNPs, short for single-nucleotide polymorphisms.
So a SNP is really any position in the genome where there is variation between individuals in the population.
Nearly all of our DNA is exactly the same from person to person. But there are about 3 million spots where a single letter difference will commonly show up. Each of those spots is a SNP. It's just one letter, and in most cases, we don't know exactly what it does.
You might have a C at one position, and I might have a T. We're both alive, we both have arms and legs and noses and brains and totally functional. But those subtle variations are what make you and I unique and different. But it's also what predisposes individuals subtly to these common diseases.
So which SNPs do I have? And what can they tell me about my health? To find out, the DNA I provided in all that spit is squirted onto a silicon gene chip, which holds millions of tiny fragments of reference DNA to compare with mine. Wherever a strand of my DNA fits with the sample DNA, the two will link and light up, revealing exactly what letters I have in certain spots. So we're looking at my actual...
This is your actual genome. And what you can...
As represented on this chip.
Correct.
Mm-hmm. The bright white spots reveal my SNPs. Now, what Navigenics will do is compare them to the SNPs of people with certain diseases. If I have particular SNPs in common with them, then I'm more likely to get those diseases. Stefan believes that if my SNPs put me in the group more likely to get a disease, like diabetes, for example, I'll be more motivated to adjust my lifestyle and lower my risk. It's a nice idea, but will it work?
I don't think we should tell anyone that this will help their health, because we really have no idea. So the thing is that
Geneticist David Altshuler has serious doubts about the current value of this kind of DNA test.
It might help their health, it might do nothing to their health, and it might hurt their health.
But how could a DNA test hurt your health? Well, take the case of Alzheimer's disease. There's a gene that works inside the brain, and one version of this gene can lead to Alzheimer's disease in old age. If you look at this SNP in your DNA, and you have a G right here, there's a strong chance that you'll have that dangerous version of the gene. If you have 2 copies of the G, one on each chromosome, your risk of getting Alzheimer's, though not 100%, could be 10 times higher than average. So what would you do with news like that?
There's a minority of people who it won't bother them at all what their genetics are. They just live their life and say, I don't care. But I worry about the other side of the bell curve. Those who get genetic information, they look fine when they get it, and then they go home and freak out, or in some cases even, may commit suicide.
Even James Watson, who deciphered the structure of DNA and recently had his entire genome sequenced, did not want to know if he had the high-risk version of the gene, called ApoE4. Alzheimer's expert Rudy Tanzi can't blame him. Tanzi strongly discourages most people from testing for ApoE4.
It needs to be emphasized that whether you inherit 1 or 2 copies of ApoE4, you are not guaranteed to get Alzheimer's disease in your lifetime. So it just doesn't make a whole lot of sense to do an E4 test for Alzheimer's disease, given that it's not a reliable predictor for the disease.
This Alzheimer's test is so controversial, some genomics companies won't even offer it. But Navigenics does. I thought long and hard about whether I wanted to hear my status, even if I couldn't do anything about it. And decided to find out. I'm a little worried, but I kind of think I'd rather know than not know, because I can use that information to adjust how I might live going forward. Finally, the moment of truth arrives. Have you seen them yet?
Well, we review everything.
That's a yes. For about 10 diseases, Alyssa Levin, Navigenics Director of Genetic Counseling, gave me two numbers. First, an American man's average lifetime risk of getting a disease. And then, my genetic risk, based on how many good or bad SNPs I had. So I'm looking here, obesity, 22%. They say my risk for becoming obese is 22%, while the average risk for males in the population is 25%.
So you're just under the population average.
I'm a little less likely.
A little less likely based on your genes.
So heart attack.
That's about 52%.
52, and I'm coming in
at about half of that.
About half of that. But what about Alzheimer's? Do I have a G in those dangerous spots? Nope. The test said I had an A on both chromosomes. So this tells you I probably don't have that gene.
I have to say, this is a pretty stellar profile.
But what's this? Okay. It looks a little high. And so prostate cancer, 17% in the general population, and I come in at 20%. Right. So remember, these SNPs cannot tell you for sure if you'll get these big common diseases.
First of all, they're only clues. We don't know what they mean. We don't know all the mutations in those genes. We certainly don't know how they work biologically. We have no clue what they mean clinically. We have no idea how they would affect the treatment you should take.
Even if we could pinpoint all the genes that contribute to the big common diseases, that still wouldn't be enough. We know from studying identical twins, genes can't be the whole story. Even with the exact same DNA, one twin might get cancer, the other won't. And we still don't know why.
For the majority of age-related diseases that, challenge healthy aging, we're still in process of figuring out the genetic factors, never mind figuring out how they interact with the environment or even what the environmental factors are. These are early days.
For now, skeptics say these tests may be no more predictive than a good family history. But what if we could find a cheap way to read not just SNPs, but the entire genomes of many people, as they're trying to do here at Harvard Medical School? If we combine those with personal histories, intimate details of our habits and behaviors, could we get closer to overcoming the big killer diseases?
The more sequence we capture in people with disease, till it's complete and different numbers of people and different populations, we will, I am entirely confident, learn about disease and human biology, and we'll learn about human history. We're going to learn about ourselves. So knowledge itself can be challenging.
How do you identify a true masterpiece? We don't usually think of a great painting as something that can be reduced to numbers. Got a couple more threes right over here. But a few scientists might disagree. They say that if you take away the color and break down a painting brushstroke by brushstroke, you'll find a complex pattern of numbers. A pattern unique to every artist. And, as correspondent Chad Cohen reports, it could provide a crucial clue if you're trying to tell the real thing from a clever forgery.
Deep in the storage vault of the Van Gogh Museum in Amsterdam, a precious canvas is being photographed. The Reaper, painted by Vincent van Gogh in 1889, has the thickly layered paint and bold curves the artist is so famous for. But are we looking at the real thing? Because there are two Reapers in this vault. One, as you might expect, worth millions. The other, a copy, worth no more than the oil and canvas it was painted with. Can you tell the difference? It's something hundreds of collectors ask the Van Gogh Museum to do every year, convinced they own a masterpiece by the great artist.
We get many questions for expertise here. Our opinion is asked all the time, and there are many fakes that have been around for decades.
This is one of 30 fake Van Goghs created by a German art dealer named Otto Walker in the 1920s. He came up with the ingenious method of priming his canvases with white lead paint to build up the thick layers Van Gogh is famous for. Then added bright colors on top. For a few years, the Walker forgeries managed to fool the greatest Van Gogh experts in the world.
It is a problem, but it's a problem for any renowned artist, because money, of course, is very tempting.
This is serious money. $50, $70, $80 million a piece for some of the best-known paintings. Even the Van Gogh Museum was fooled into buying a fake back in the 1970s. It's a 19th century painting called View of the Eye, Amsterdam's inner harbor. It wasn't originally intended as a fake. But years later, someone forged Van Gogh's signature onto the canvas. So how do you determine if a painting is the real thing? In May of 2007, the Van Gogh Museum explored one of the latest scientific methods of art analysis by hosting the first international workshop on computer image processing.
These images are represented in terms of numbers.
Eric Postma directed the technical presentation.
The purpose of the project was to look at these paintings and try to determine if we could distinguish between real and fake Van Goghs.
The computer scientists focused on one of the most revealing traits of Van Gogh's work. As the museum's head conservator, Ella Hendricks, showed us, it's the way he applied paint to his canvas.
I would tend to really want to get close and look at these, for example, these very nice white strokes in the foreground that were added at a late stage. And if you look very close, you can see these sort of trailing drips of paint as he literally scooped up the paint from the canvas, lifting his brush and then carrying the trail of paint.
It turns out that Van Gogh's brushstrokes are crucial markers not only for critics and connoisseurs, but for computers as well.
The pattern of brushstrokes is very specific and very indicative of a painter. So the first selection that you make when you start to analyze these paintings digitally is to select the level of individual brushstrokes.
The computer scientists create programs which examine a painting from different angles to determine the direction of each brushstroke. Collecting the brushstroke patterns from more than 100 paintings establishes a baseline for Van Gogh's style. But could the computer be fooled? Could an artist specially trained to copy the old masters duplicate Van Gogh's style? To find out, we commissioned Charlotte Kaspers to carefully imitate every detail of Van Gogh's brushstrokes. She's an expert in art reconstruction, using historical materials to copy famous paintings so museums can decide how to preserve the originals.
The best would have been to sit next to the painting, but, well, that was just not possible.
The Van Gogh Museum doesn't allow artists to paint in front of the originals, so she made her own photos of the Reaper.
I couldn't bring the painting home, so I had to take a picture, and when it's a digital one, you can blow it up on your computer, so that was what I did.
Knowing that the scientists would be focused on the pattern of Van Gogh's brushstrokes, Charlotte's job was to perfectly match his technique.
I had a picture next to my canvas I was painting on, and I could blow that up so I could have a close look at the paint surface of the original painting from a picture.
How do you think that trying to be him ultimately influenced the painting itself?
It's less spontaneous. I was just switching all the time my eyes from my canvas to the picture and trying to do what he did. The charcoal, I did his outlines and here the folds and the indication of the face. And then I added shadows. And then after that I did something with the dark lines. And then I added the highlights and the outlines. And I did the birds in the end. That was the last thing. It was fun to do.
We had a young artist copy one of the master's paintings. Did you get to see the
I did get to see it, yeah.
What did you think?
I think she did a remarkable job because she's made a very accurate copy of the rhythm of the brushstrokes, but managed to do it in a spontaneous way.
So do you think she has a shot against the computers?
I think it will be a challenge, yeah.
The challenge begins by turning Charlotta's copy of the Reaper into a color photograph, which is then turned into a high-resolution black and white scan. Identified only by catalog number, it takes its place beside 5 real Van Gogh paintings, creating Nova scienceNOW's test for 3 computer teams: Penn State University, Princeton University, and Maastricht University. Eric Postma leads the Maastricht team.
So here's a famous Van Gogh.
Yeah.
Van Gogh.
Van Gogh.
Van Gogh.
Van Gogh.
Van Gogh. The sower.
De zaaier.
However you want to say it. How do you begin? So you look at this, it's a beautiful painting, but you're going to strip it of its beauty, one layer of its beauty right now.
Yeah, in fact we do, because we translate the entire painting into numbers.
Postma's software scans the whole painting, identifying areas of contrast. The more contrast, like the dark tree branch against the light sky, the larger the number assigned by the computer. If the contrast is less extreme, like the edge of the sun against the sky, the number is smaller. When all the contrast patterns are combined, a statistical portrait of Van Gogh's style is created.
Well, these are genuine Van Goghs, and this is a known fake, the so-called Walker fake. The faker tried too hard to mimic Van Gogh.
And by trying too hard, that means
painting
much.
Too much. Too much, exactly.
I see.
Someone copying Van Gogh paints too many brushstrokes to get it right, and the computer identifies them as areas of higher contrast. First, the program analyzes the brushstrokes from 6 different angles, then combines them to reveal areas where they overlap. More overlapping brushstrokes show up as brighter areas. Fewer overlaps are dark. Look at this famous painting Van Gogh did of his bedroom. As the computer separates out each angle, it highlights the brushstrokes painted in that direction. Putting them all together reveals a brushstroke map of the painting. Now look at the Walker forgery. The computer identifies more overlapping brushstrokes in every direction, 10 times as many as the Van Gogh bedroom. The genuine Van Gogh paintings display fewer brushstrokes than the forgery.
If you deliberately try to mimic Van Gogh, then it's not natural anymore, and then you tend to overdo it. And this overdoing it results in high numbers, and that could be one way of detecting a fake.
I'd like to see what the zoom-in gives there.
Ingrid Dopschi and the Princeton team have also been looking at the 6 paintings in our test, searching for the fake.
Okay, so...
If you try to make a copy, you would pay so much attention to what you're doing that you probably paint it more slowly and with a more restrained hand than Van Gogh himself would have painted, and we expect that to transfer as well.
For James Wang and Jia Li from Penn State University, Charlotta's copy seemed to blend in with the other 5 paintings.
Just by looking at the pictures, they all look very Van Gogh. So that's why later we also tried a statistical modeling approach.
So you get a kind of segmentation of the painting.
All the science teams had less than a week to distinguish the fake from the real Van Goghs.
Typical brushstrokes.
I think we see more contrast in the grooves than in the ridges.
Oh yeah.
This white thing is showing through.
Oh, yeah.
Would they pick the sunflowers, the sower, any of the other 3 genuine Van Goghs? Or would any of their computer programs successfully choose Charlotta's version of the Reaper? The analysis is finished, and it's time to take her painting to the Van Gogh Museum, where the scientists are waiting.
Okay, the moment of truth. Do you all have an answer?
Yes.
Yes. We're ready to bring in the artist?
Yes.
Okay, Charlotta. This is Charlotta Casper. She painted the painting that you guys tested.
Oh, very, very nice to meet you.
Do you want to come on over and reveal it? Well, bring it on over. Don't open it yet. Bring it on over, Charlotta. All right, so what do y'all got?
687.
687. Right.
687. The one.
You guys match. That's 2 for 687?
And that's 3 for 687.
Yeah. All right, that's your final answer. You're sticking with it.
Yes, yes.
Yes.
Here we go.
All right, Charlotta, let's see.
Yes,
Okay, let's
yes,
see.
yes, yes.
Wow.
Oh,
yes,
687, the Reaper.
Yes, got it.
So how long did it take for you to finish the painting?
I worked 2 days on this one.
How long did it take you to analyze the painting?
Oh, we took 2 days too.
In fact, Van Gogh himself completed many of his most famous paintings in 2 days or less. And they've kept art collectors and experts busy for more than a century. They're not there yet, but computers will ultimately speed the process along.
The most valuable thing about these techniques is that they can help art experts. You just can scan over the entire painting, and they do it in a completely unbiased way. And that's the great advantage of computers.
Every creature on Earth does the same thing. We take in oxygen and then give out carbon dioxide with the same breath. And while we're putting out CO2, trees like this one and other plants are sucking it right up. They need it to survive. But with worldwide population growth and increased fossil fuel consumption, we're now putting out more CO2 than our trees and plants can absorb. And since CO2 is a greenhouse gas, there are fears that all this carbon dioxide is heating up our planet. For some, the solution is obvious. Correspondent Peter Standring met up with some inventors trying to design a very green machine, one that can make like a tree.
In a warehouse on the outskirts of Tucson, Arizona, Professor Klaus Lackner has come up with an idea even he admits is a bit fantastic. He's attempting to compete with Mother Nature.
We're trying to mimic what a tree can do, and these are the leaves of that tree.
Mimic a tree?
Some people would say this can't possibly be done, but then on the other hand, every tree can do it.
Every tree, in fact, every leaf, is like a tiny factory, taking in carbon dioxide from the air and using it to make the energy it needs to survive. In the process, it releases the oxygen that we need to live. Klaus's version of a tree also pulls carbon dioxide out of the air, not for its own survival, but to help us fight global warming. Sounds pretty incredible? Well, so is the way he came up with the idea. It all started a decade ago, when his 12-year-old daughter Claire came to him for advice.
When I started to think about this problem, I was looking for ways of doing experiments. And just about that time, Claire came to me in the study at home and said she is looking for an experiment to do for her science fair.
I was in middle school and I had to do a science experiment for my science class, and so I talked to my dad about various ideas and he suggested this.
And I said, why don't you pull CO2 out of the atmosphere?
Pull carbon dioxide out of the air? A tall order for a little girl. But as the daughter of a renowned scientist, Claire already knew about global warming. She understood that when sunlight enters the atmosphere and strikes the Earth's surface, some of it is reflected back towards space in the form of heat. Greenhouse gases like CO2, carbon dioxide, work like a chemical blanket to trap heat and keep the planet nice and warm. But the increased burning of fossil fuels was generating so much carbon dioxide that our planet's temperature appeared to be rising at an alarming rate. But how could you just pull CO2 out of the air? Claire had an innovative idea.
I went to the local pet shop and bought a fish pump.
All right, there you go. Have a good day.
You too.
I filled the test tube with sodium hydroxide. Next, I attached the fish pump to the test tube, turned it on, and ran air through it all night.
As Claire slept, her experiment was hard at work. The fish pump was forcing air containing a small percentage of CO2 into the test tube. CO2 is an acid like vinegar. Sodium hydroxide, the liquid in the test tube, is a base, kind of like baking soda but stronger, a lot stronger. It's made of lye, the nasty stuff that cleans out your drain. When acids and bases meet, they not only attract, but they bind to each other. It's called an acid-base reaction.
So the carbon dioxide binds to the sodium hydroxide and leaves the air.
Claire succeeded in capturing carbon dioxide straight from the air, won a prize at the science fair, and changed the course of her father's life.
I was surprised that she pulled this off as well as she did, which made me feel that it could be easier than I thought. The first sketch I made ended up looking like a tuning fork or a goalpost with Venetian blinds.
A far cry from Mother Nature's design.
The first reaction of most people is, why take CO2 out of the air where it's more dilute than in any other place? Clearly it must be easier to get it out of a power plant. But not all of the CO2 comes out of a power plant.
A lot comes from cars, trucks, and airplanes burning fuel. Once in the air, CO2 is very dilute, making the idea of capturing it sound close to impossible. If Klaus was going to make this far-out idea a reality, he was going to need some practical advice.
You look at Claire's experiment, what she had is a test tube.
So he went to the Wright brothers, not Orville and Wilbur, but project manager and engineer Alan and Burton, another set of brothers who, like their namesakes, don't shy away from a challenge.
The Wright brothers were able to look at a bird in flight. So they knew it was possible to fly. So does the fluttering of the leaf. Klaus and I will look at a tree and say, well, you know, that tree is capturing carbon dioxide out of the air. We know it can be done. You've got to figure out how to do it.
Not just in the laboratory with a tiny fish pump and a test tube, but on a global scale. In 2004, they form a private company called GRT. But the transition from a child science fair project to the first synthetic tree is filled with obstacles. One in particular could stop them dead in their tracks. Their tree needs electricity to run, and whenever you produce electricity, by burning oil, gas, or coal, you also produce carbon dioxide. It's called an energy penalty. If their synthetic tree produces more CO2 to run than it can capture, well, what's the point? A delicate balancing act begins. For every choice the team makes, there is a price to pay, an energy penalty. Can they somehow reduce the amount of energy they use?
We needed to come up with a shape where you don't have to have an aquarium fish pump driving all the air through the system, but to have the wind just deliver the air and pass it through the collector.
It all comes down to geometry. What is the size and shape of the perfect synthetic leaf? One that can remove the most CO2 from the air. To find out, they construct a wind tunnel to study how air moves around and through a variety of surfaces.
The easier it is to get air through, the more CO2 we can collect.
We tried an array of strings. We tried screens. We tried vertical plates of solid material that were smooth. We tried vertical plates that had a knobby surface.
With each attempt, they measure the air pressure in the wind tunnel. A drop in pressure means the airflow has stopped, and that sample has failed.
19. So this is not the answer.
It takes a year to find a shape that lets enough air pass through.
Perfect.
It turns out to be long, flat sheets.
The air would move through with very little resistance. It worked well.
So, air can move through their man-made leaves, but how will they capture CO2? At first, they follow Claire's lead, coating the leaves with sodium hydroxide. The chemistry is sound, but it's a nasty business.
It will be a much tougher job for us.
Sodium hydroxide is great to prove that it can be done, but it has so many disadvantages.
Sodium hydroxide is a very corrosive material. It's not a good idea to get it on your skin. It's very harmful if you were to get it in your eyes. As a practical matter, trying to build a machine that works on sodium hydroxide would force us to use very expensive materials, would drive the cost up significantly.
The guys decide to abandon the idea of using sodium hydroxide when they make a startling discovery: this material. Now, exactly what this material is, the guys aren't telling.
What is it? Do I have your attention? We can't tell you.
Turns out this is where science and commerce collide. Its true identity is proprietary. That is, until their patent comes through. The team claims that this engineered fabric attracts CO2 just like sodium hydroxide, but with none of the pitfalls. Here's how the system works. The 9-foot synthetic tree opens its doors, letting air flow through its leaves, which, thanks to their mystery material, readily absorb carbon dioxide. The leaves are then sprayed to wash the CO2 away for storage. The process does use electricity, but in the future they hope green power will make the device even more energy efficient. Still, one big question remains: what to do with all that CO2? One option lies miles from civilization. Since 1996, a Norwegian oil company has gotten a lot of practice getting rid of CO2 by pumping it into an aquifer deep beneath the North Sea. The process is called carbon sequestration. But could the carbon leak out? If so, what effect would it have on marine life?
Once you've got enough gas under there and it's leaking out, it could become a very serious problem.
And how much CO2 can they put down there anyhow?
I believe in the long term, underground injection will not quite have the capacity we are looking for. So I am looking at another process, which I refer to as mineral sequestration.
There's a perfect example of it in New York City, on the campus of Columbia University, underneath a bronze statue of the school alma mater.
She is sitting on this pedestal of serpentine rock. This serpentine has absorbed CO2, probably out of rainwater. It's known as geological weathering, and if you wait long enough, that's what will happen to all of the CO2 we make.
But it takes hundreds of thousands of years for Mother Nature to pull off geological weathering, and we don't have that long to wait. So Klaus is trying to figure out a way to speed it up in the lab. As for his tree, he now has a working prototype, but many questions remain unanswered. Like, how well will it survive the elements? And who's going to pay for it? Is Klaus's tree too fantastic to be real?
You could have said that about the Wright brothers and Thomas Edison. I can't sit here and tell you now that this is going to work. I can tell you now that it would be a terrible mistake not to do the research to find out.
I believe that it is impossible to stop people from using the fossil fuels, so we need to develop technologies which allow us to use them without creating environmental havoc on the planet.
We are, as a world, changing the climate and changing the Earth, and we need to understand how we're changing it and understand what we can do either to fix it or to control how we change it.
I think that I shall never see a poem lovely as a tree whose hungry mouth is pressed against the earth's sweet glowing
A lot of scientists, just like a lot of people, secretly wish they were rock stars. But really, how hard could it be? Our love was like a supernova in the nebula of my soul. But now, I find your heart is just a big black hole. Well, maybe it's harder than it looks. But in this episode's profile, you'll meet a scientist who has a much better shot at becoming a real rock star. Don't believe me? Check this out. 32-year-old Pardis Sabeti is a member of the rock band 1,000 Days.
The deer and the heat under the lights all over, they leave us here.
Singer, bass player, and songwriter, she's a rocker by night. Come morning, Pardis plays a different tune.
I'm a geneticist with an interest in infectious disease.
This is her stage by day, complete with its own dressing room, and of course, her instrument of choice. In 2001, Pardis used her instrument to make a major breakthrough in genetics. She was combing through the human genome, trying to track natural selection.
Well, the process of natural selection is one of the most fundamental driving forces of evolution.
I'm looking for all the things that are beneficial in the human genome. Everything that I do is based on a very simple principle. Things that are beneficial will spread through populations very quickly.
Think of it this way. In natural selection, our DNA, our internal blueprint, constantly mutates. And some of those changes help us to survive in our given environment. Those beneficial changes will be the ones that get passed down to future generations.
If an advantageous mutation should occur, something that might protect you against a disease, for example, you'll leave more offspring. And your offspring will leave more offspring.
And sometimes, a genetic mutation will allow you to take advantage of a new source of food. For example, cow's milk. Thousands of years ago, most adults couldn't drink milk without getting sick. But after cows were introduced to Europe, people with a particular mutation that allowed them to drink milk safely could take advantage of this new food. They had a better chance of survival and passed that milk-drinking gene on to the next generation. Today, 80% of European adults can drink milk. That's a genetic brush fire. Pardis figured out how to spot brush fires like these in a genome. She came up with an algorithm to compare genetic codes among hundreds of different people around the world and identify the genes that have become very common very fast. A sure sign of natural selection at work.
The cool thing about what Pardis did was figure out what to look for in the first place. Figure out how to sift the haystack to find the needles.
And this idea has created a little brush fire of its own.
And Pardis's idea has been picked up by many, many groups now. It's now considered a routine tool for scanning the human genome to understand evolution.
Pardis is a graduate of MIT and the Harvard Medical School. In 1 year, she received not only a 7-figure research grant from the Bill and Melinda Gates Foundation, but an honorable mention in Billboard's World Song Competition. A female rocker who was born in Iran and is now a genetics professor at Harvard. Wait, let me repeat that. A female rocker who was born in Iran and is now a genetics professor at Harvard. Pardis is her very own mix of a new generation's sensibilities with the old generation's credentials.
If you do what you love, things happen fast. And I think that I don't manage my time well. I don't think I do anything special. It's just I stick around things that I love.
She learned that at an early age. Okay, maybe not this early, but her biggest lessons in life came from a unique family experience. When Pardis was only 2 years old, her family was forced to leave their home in Iran because of the revolution. They found refuge in the United States, but they had lost everything.
You establish a life in one country, and then you get to a new country to sort of like start over, learn a new language, and get going. So it took a while.
All of this uncertainty meant that throughout her life, there were only two things Pardis could count on: herself and her family.
And we were always together, which was great, and so then you felt safe.
Parisa is Pardis's older sister, appointed playmate, and caregiver since birth.
From the moment that my sister came home, my mom made a big effort to say, "This is your sister, take care of your sister." The two of us were always very, very close.
After her family finally got its new life in America on track, Pardis was in the 9th grade when her father was in a car accident that nearly killed him.
He shattered both of his legs. It was an 18-hour operation to piece his legs back together. Months and months and months in the hospital. They really didn't think he'd ever walk again.
Her father made a full recovery, but this experience changed Pardis's life. She apprenticed with the doctor who performed the operation, which led to her decision to study medicine. And her father's example provided a lifelong lesson.
As long as I have a heartbeat, I'm fine. So I just do what I love, and I do it the best that I can, and if it all goes away, I'll just start over. You get this added drive because life is so precious.
These days, Pardis has taken up the challenge of fighting malaria, a disease that infects at least 300 million and kills 1 to 2 million people each year. She's traveled to Africa to study how the malaria parasite interacts with its human hosts. And, just like the human genome, the genome of this parasite, spread by mosquitoes, has now been mapped. Pardis is searching its genome for signs of natural selection in the hopes of understanding how this parasite develops resistance to the drugs we use to fight it.
The same types of methods I use to study what are the things beneficial in humans that allow it to continue to survive and stay on this earth, we can do the same thing with malaria.
This approach that Pardis is using is bound to lead to breakthroughs.
What's wonderful is Pardis has no boundaries about what she'd like to do, what she's interested in doing. Her problem will be how to choose which of those she can fit into a 24-hour day.
And while the future is anybody's guess, Pardis insists she's a scientist through and through. As she says, it's much cooler than being a rock star.
You get these moments of thrill. There you are at 3 in the morning and you know something about how we evolved that nobody else in the world knows. It's a thrill of discovery. You make this breakthrough and you find something. It's this wonderful, wonderful scavenger hunt when you got to the end. Like, it's just so great to be a scientist.
As I am, it's time that I just fit, and I'm coming up faster than I ever have before. I can't hold my breath, and I'm coming up faster than I ever have before. You know, this time I couldn't.
And now for some final thoughts on bad news. Many people fear new information that might contain bad news beyond their control. But to fear bad news by hiding from it forfeits any opportunity to solve the problem. Suppose the enterprise of science shunned bad news. We would never cure disease or mitigate disaster. We would crouch with our head in the sand, ceding our fate to forces we imagine to be beyond our control. Take killer asteroids, for example. Without a space program, we could pretend they're not there, or we might run away from where they might strike. But I'd much rather learn all I can about these objects and then figure out a way to deflect them. Even worse than bad news for some people is uncertainty. Not knowing something for sure makes them uncomfortable. They just have to have the answer, the exact answer. But to the scientist, Uncertainty is a call to the wild. That's where data are in greatest need of improvement along the way to fully develop theories and experiments that have shed their shrouds of uncertainty. For some cases, especially your health, you can actually influence the uncertainty yourself. If you wear a seatbelt, your chances of surviving a high-speed car accident are much higher than if you don't. And if you exercise and eat well, you can stave off diseases that you might be genetically prone to contract. So, bad news, uncertainty, I say, bring it on. To a person engaged in discovery, all information is good, even when it's bad. And that is the cosmic perspective. And now, we'd like to hear your perspective on this episode of Nova Science Now. Log on to our website and tell us what you think. You can watch any of these stories again, download audio and video podcasts, hear from experts, and much more. Find us at pbs.org. That's our show. We'll see you next time.
Educators and other educational institutions can order this NOVA program for $24.95 plus shipping and handling by calling WGBH Boston Video at 1-800-255-9424. NOVA is a production of WGBH Boston.